Right, we should be live, so welcome to the B1 exam revision live stream. As I said in the little preemptive bit about half an hour ago, if you've got a question, the way you're going to be able to ask them this time, rather than putting them in the comments, is to email this address, communications@folkestoneacademy.com. If you email that, this address here, we will try to get your questions up when we get to a break. What I'm going to be doing is I'm going to be doing about half an hour of me talking you through a few of the key ideas from B1, ones that I either know come up or are difficult topics that we find hard to get. And then after about half an hour I'll be stopping, going through some questions that have come in, and then moving on to another sort of half hour after that. We'll get through what we can and we'll see how it goes, and hopefully this will be useful to you watching. So, B1, exams tomorrow. Okay, there's lots of topics that could come up in B1. We've got healthy eating, pathogens, nervous response, biomass, energy and biomass, decay, genetic variation, evolution, genetic modification, amongst others. And I'm sure you've been doing lots of revision on these topics already since the exams tomorrow. So this is kind of just a last minute refresher. I'm not going into much detail on many of the topics because I don't have time to do it, but what I will be doing is going over the basics of lots of different bits, and hopefully when you get to that exam tomorrow they'll be fresh in your head, and you'll be able to, like I say, just have these little tips that will pop up and you'll remember. So really it's just little tips to remember to help you pass tomorrow. And by pass we mean get whatever grade that you want to get out of GCSE Science. That's what we've been doing all year. We're trying to help you through this. Okay, so first topic we're going to be looking at is nerves. And I've talked a lot about nerves with my class, but if you're not in my class or you're still a bit unsure, then I'm going to go through it again, because it's one of the topics that does come up a very large amount. I'm going to talk specifically about synapses in the next bit. So I'll talk specifically about synapses, and we'll come to what those are if you're not sure at this point. This is a sort of nervous response. Now, if you can't see it, don't worry. Okay, the ideas are sound, and I'm going to be going through them anyway. Your body has what's called receptors, and receptors are things that pick up bits of information. They're usually, like when you're learning it earlier on in your school life, you probably call them your senses. So you've got your eyes, your ears, your nose, your tongue, your skin. All of these have receptors in them, and they pick up different signals. So your eyes respond to light. Your ears respond to sound, your nose responds to smell, and so on and so forth. Okay? They're the receptors. So they pick up what's called a stimulus. A stimulus is just a change in the environment that they detect. So they detect a change in the environment and they respond to it. And what they do is they actually send a nervous signal down what's called a sensory neuron. So they send that signal down a sensory neuron. If you're not sure of this process, so I'll write it up here because it is quite small, the board. So you've got receptor, which picks up the signal, and that goes to a sensory neuron. Okay? That signal then gets passed down along as an electrical impulse, and that's very important. Down the sensory neurons, or the neurons, there's sensory impulses that go down. And those electrical impulses, they move down until they reach the central nervous system. The central nervous system, or CNS, central nervous system, is the coordinator, and it coordinates all your body's responses to different changes in the environment. Okay, so for me, an example would be I see the pen. That's the stimulus. Okay, it goes into my eyes, which are the receptor, sends a message down a sensory neuron to my brain. My brain then decides it wants to pick it up. So what happens next? That sends a message down a motor neuron. So the central nervous system sends down a motor neuron. Okay? And that's actually an electrical signal as well that gets sent along. That motor neuron actually transfers the signal to what's called an effector. So it has an effect. And what you need to be very clear on effectors, or effectors, is there's two types that you need to know about. Muscles that cause movement. So in my example before, when I'd sim the pen, sent a message down a sensory neuron, gone to my brain tells me I want that pen, sends a signal down a motor neuron to the muscles in my arm, which cause me to reach forward and pick up that pen. Okay, that's one type of sensory response. Okay, so that would be a normal nervous response, the pathway it takes. You can answer exam questions with a flow diagram. That shows the actual nervous response in a flow diagram. You can use that to answer questions. You don't have to write out long sentences for science questions. The only one you actually have to do that for is the QWC, the 6-mark written question. Okay? All the others you could do a diagram such as this, if you could remember that, or a flow diagram such as the one I've got over there. Now, I've talked about the effector being a muscle, and that causes movement, so muscles cause movement. You can also have an effector that's a gland, and glands can be in various places of the body, but they release hormones, or they release enzymes into the body. And that can have an effect on the body. So another type of nervous response would be a faster one. This one that's actually shown is actually a reflex response. Okay, and this reflex response happens quicker than a normal nervous response because it doesn't involve the central nervous system, it doesn't involve the brain. It actually goes through what's called a relay neuron. And a relay neuron is just a short neuron that links the gap between the sensory and the motor neuron. So it sends the message a lot quicker, and reflex actions are usually to protect you from harm. Okay? So if you touch a hot pan, okay, if you're touching a hot pan, the skin receptors pick up that signal, send a message down a sensory neuron. You don't want to think, oh, that pan's hot, it's going to burn me, better take my hand off, because by the time you've thought all that, you're going to have caused serious damage to your hand. So instead, the message just goes through a relay neuron, and it instantly sends it straight to a motor neuron, which causes the effect, moves your hand away. So that's called a reflex action, and happens through a relay neuron. So it's different, doesn't involve the central nervous system, doesn't involve the brain, happens a lot faster. Okay, so that's the only two differences in the responses. Okay? So that's how a normal nervous response sort of takes place. Okay? You can go from a receptor to a sensory neuron to the central nervous system to a motor neuron to an effector, a muscle or a gland. Now I said I wanted to come back to a really important point in this that gets asked a lot. Okay? It's the gap, or sometimes called the junction, between two neurons. Okay? What can happen there is very important in GCSE. I see it come up a lot, and it's actually a tricky little bit. So, to show you that, this is actually a close-up of that junction, the bit in between the two neurons. It's called a synapse. Okay, that's the name for it. It's called a synapse, and it's actually a little gap, a little space between them. And in between there, a different signal is sent. So, say this was your sensory neuron that goes down, and it ends in this gap, which is called a synapse, and this is maybe the relay neuron that goes away from it. In between that gap, it's not actually an electrical impulse or electrical signal that's sent. So electrical signal arrives down here, and then there has to be a chemical transmitter that bridges that gap. So it actually sends out chemicals from one end, from one side of the neuron to the other, where they get picked up and they get detected. When they are detected, those chemicals, it tells another electrical impulse to start firing and move along this neuron. And then that could be a motor neuron, or could just be the relay neuron, whichever. Doesn't matter. It's the fact that there's a gap between the two neurons, and chemicals are used to send the signal across that gap. So electrical impulses in the neurons, and then chemical signals between them in the synapses. It's a really important bit, and it comes up a lot actually. They ask you quite a few times about it. Sometimes very simple questions, such as what form does the signal take between a synapse, and sometimes more in depth, where you actually have to describe chemicals being released based on the electrical impulse. The chemicals are picked up the other side, and that starts a new electrical impulse the other side of the neuron. So that's nervous response, okay? That's one of the topics, a hot topic for B1, and a bit about synapses, which goes into a bit more detail about it, okay? Another topic that I don't think gets covered enough, and often students still don't quite get, is evolution. Okay, there's two theories of evolution that you need to be aware of. One of them, Lamarck, is actually fairly simple when you describe it, and Darwin's is actually much more complicated. You need to be aware of both. Okay, you need to be aware of both of these types of evolution. Lamarck's theory, we're not going to go into too much detail on because you don't get asked much about it. You just need to be aware and have an understanding and be able to recognise when Lamarck's theory is being suggested in a question. His theory suggests that animals, if they used a certain characteristics, could change that characteristic in them. And then when they change that characteristic, they could pass that on to their offspring. An idea it would be like, if this was true, it means I would be able to build up my muscles, get even buffer than I already am, and then if I had children, they would also be like have muscles. Okay, so passing it on by that, by using something enough. A classic example is giraffes. So where giraffes used to have short necks and they stretched their necks to reach up to trees, and after a number of times of stretching their neck, they actually grew their necks and passed that on to their children, to their offspring. His theory says that we all evolved from worms. We all started from worms, and by sort of using different characteristics, by using different appendages and things like that, we actually changed into the complicated beings that we are today. It's since been proven that he was wrong, but at the time he suggested it. Okay? People didn't like it because of this idea that we were descended from worms. They felt that made us too lowly. Also, for both theories of evolution, it was difficult to get them accepted at the time because a lot of people had religious beliefs. You can't just say, if it says why were these theories not accepted at the time, you're not allowed to just say religion on its own. You have to say why that means it wasn't accepted. So therefore, people had religious beliefs, so did not believe that we evolved from worms, that we were created by God. You have to put a bit more detail into it. Darwin's theories, we have to know how they work. Okay? His theory, he suggested that animals changed by adapting to their surroundings. So their surroundings caused them to be different, and over a long period of time, it actually caused species to evolve and to change. Not by using a certain characteristic, by a process of what's called natural selection. And that's what you need to know, how natural selection works. Again, I see quite a few answers from students who just say, explain Darwin's theory of like evolution, and they'll just say, oh, natural selection, expect to get marks. You have to describe what is natural selection. You have to go into the details of what that means. So to do that for you, I kind of tried to break it down into stages that would get you marks in exams. Because a good exam tip is, if a question's worth 4 marks, you need to make 4 distinct points about it. And you can bullet point your answer. Never forget that. You can bullet point, and if you've got separate bullet points, and you've got 4 of them, then you have a chance of getting those 4 marks. Okay? So, evolution by natural selection. How does it work? Okay? Within a species, or with a species, there is variation. That means that we're all different. Okay? We're all humans, and apart from the obvious gender differences, even within the same genders, there is variation. No 2 people are alike. Okay? And that's the same for all species. There's always slight differences in us. That's partly due to we have inherited characteristics from our parents, but it could also be due to mutations in the DNA. And this is where natural selection really sort of came forward, or this idea came forward. If there's a mutation that caused the animal to be significantly different, that could be an advantage. And if we go back to my example of giraffes, which again they do like quite a lot, some of the giraffes were born with long necks and some with short necks. And they were all living together, and they were all one species at the time. Those mutations that caused the giraffes to have a long neck, or what was the ancestor of a giraffe I should say, to have a long neck, actually made it more successful at living than other of the same species. So other giraffes, although they weren't actually giraffes at the time. But those characteristics, the long neck, made it more successful. And you should, if you can, say why you believe it was more successful. For example, it was more successful at finding food in the example I'm using for a giraffe. because it could reach the trees and reach the branches of the tree and eat the leaves from higher up than the short-necked giraffe, so had access to more food. Okay? So that makes it more successful. And if you see, I made 3 points in my answer now, so if this was a 4-mark question, I'd be well on my way to getting those 4 marks. The successful characteristic that it has actually makes it more likely to survive. And if an animal's more likely to survive, it's more likely to reproduce, because that's what all animals essentially is their end goal. I'm talking about animals, but it can also take place in plants as well. So if an organism is more successful, it's more likely to survive long enough to reach maturity, to reach a point where it can reproduce itself. And that final point means the advantageous characteristics, the long neck in the example I'm using, means will be passed on to its children and its offspring. So essentially, to summarise, all the giraffes or ancestors of the giraffes that had short necks died out because they could not get the food that the long-necked giraffes could get. And when food became scarce, okay, the long-necked giraffes survived and the short-necked giraffes died, and that meant that all of the giraffes that had long necks could reproduce with each other, led to children with long necks. And soon, the short-necked giraffe, the ancestor of the giraffe, actually disappeared and a new species was created. They talk about this a bit in B2. Generally, when it's in B2, you have to talk about what's called speciation, but that doesn't come up so much in B1, so I'm just going to leave it out for now. You still need to be able to describe this process. So you don't need to be able to describe the marks in detail, you just need to say that if an organism uses a characteristic enough, it will change that characteristic. Darwin's, on the other hand, was this idea of natural selection, variation within a species caused by mutations in the DNA. Some characteristics make animals more successful, leads to them being more likely to survive and therefore reproduce, and then those characteristics get passed on to the children. Okay? So that's what we're looking at in terms of evolution. Okay? We haven't had any questions come in, so do make sure that you email in. The link is underneath the live stream. You should be able to see it. It's communications@folkestoneacademy.com. Because there's been no questions come in so far, if a question does come in, then I'm highly likely to be able to answer it when we get to the little session. If we don't, I'll just carry on through, and hopefully you're just getting all the good information from it. But if you are unsure of anything I've gone through, please do email in that address, okay? Tied into evolution, and the next topic I want to talk about is genetics and inheritance. The basics of genetics that you need to know is that the smallest unit of inheritance is called a gene. Okay? And those genes are small sections of a longer molecule called DNA. And this long molecule is actually like a strand, like a thin piece of string. And when you coil that thread up into a little sort of shape, it becomes a chromosome. So it's actually a chromosome. So chromosomes are made of coiled DNA, and a section of that DNA is called a gene. Those chromosomes are found inside the nucleus of cells. Nucleuses, or the nuclei of a cell, is the most important bit in some ways. It contains all the information needed to make the cell what it is. And that's because the chromosomes are found there, containing the DNA, containing these sections that are genes. Okay? So that section, that gene, that controls a certain characteristic. So based on that combination of genes, it actually leads to you having different characteristics, and that's how inheritance works. There's actually, you have to know a bit about what's called inherited factors, and inherited factors are facts about you, characteristics that you solely inherit. So your eye colour, you get that from your parents, okay? Natural hair colour we could use, but I'll come to hair colour in a second. That's a bit more complicated because there's aspects to it. Your sort of baseline metabolic rate, how fast your metabolism works, which is the rate of chemical reactions in the body, that's inherited. But there's also things called environmental factors. Okay? Environmental factors, which can be asked about, they're things you can change about yourself based on the environment. So hair length, you can cut your hair, okay? That can change. There's nothing that genetically that determines how long your hair is going to be. What clothes you wear, that's an environmental factor. You choose to do it, so you've changed that. what your hair colour, I said I'll come back to it, you can dye your hair, so you can obviously change that, so you could argue that was an environmental factor as well. Okay? Piercings or scars, all these are environmental factors. Some characteristics can be controlled by both, so your weight, for example. Your weight is probably slightly determined by your parents, but you can obviously affect it by your diet. So if you eat lots, your weight will increase. If you eat a balanced, healthy diet, your weight will probably stay about what it should do. If you exercise regularly, you may be losing weight. Okay? So that's controlled not only by your genetics, but by environmental factors as well. So by the both. Okay, will change your weight, for example. So this bit, you need to know the order of these things. So inside the nucleus you have chromosomes. Chromosomes are made up of DNA, and a section of DNA is the gene. So that's the sort of size order of things. Chromosome, DNA, genes as the smallest unit of inheritance. And you need to know they can decide certain characteristics within yourself, like eye colour, for example. Okay? Gender is another. You need to know the two methods of reproduction. So reproduction, there's two main methods you need to know. Well, there's only two methods, I should say. There's sexual reproduction. And sexual reproduction is the joining of gametes. And gametes is just the posh word, the scientific word for sex cells. Sex cells being the sperm in the male, and egg in the female. Okay? Sexual reproduction involves the sperm and the egg fusing together to make a fertilised egg, which will then go on to divide to become a small bunch of cells, which is called an embryo. Okay? That embryo has different genetic code based on both parents. Okay? So it actually leads to genetic variation, because half of the genes have come from the mother and half have come from the father. Okay? And because of that crossing over, it leads to variation. An advantage to this process is that that leads to evolution. Because of that variation, as I mentioned earlier in natural selection, it leads to species being able to evolve, to change over time. So that's actually an advantage. The disadvantages to sexual reproduction are that you actually have to find a mate, and not every species is good at doing that, and therefore can suffer and won't be able to reproduce. You could think of pandas. Pandas, they're having difficulty in captivity getting them to breed, and so sexual reproduction is difficult for them. They're not actually producing enough, reproducing themselves enough, and they're actually going extinct because of it. The opposite to sexual reproduction, the exact opposite, is asexual reproduction. And I say it's the exact opposite because that's how you need to remember it. Asexual reproduction, opposite to sexual reproduction. So it involves only one parent. Okay, so all of the genes, all of the genetic information comes from just one parent. Because of that, it actually results in identical copies of that parent. You could call them clones. Now, most animals are not able to do this. Okay, they need this sexual reproduction. They need the fusing of the gametes from the male and female sides. But bacteria can do that. They just divide and reproduce themselves. And plants can actually do that. So some plants, well, all plants in fact, have male and female parts to their flowers, and they can actually fertilise themselves, and that actually leads to asexual reproduction. And that's an advantage because they don't need to find a mate, and it can happen very quickly, and they can actually reproduce themselves when they're not even near any other plants. So there could be no other plants in the area, and they're able to reproduce themselves individually. So that's an advantage to a plant. Disadvantage to it would be, though, exact opposite to this. It doesn't lead to variation. So that bacteria won't be very good at evolving, and neither will the plant be good at evolving. Plant's probably a better example. And it won't be able to change itself. It won't be able to evolve because there's not this variation. So there's not this, if there was a disease that affected one of them, it would affect all of them because they would all be exactly the same. So that's a disadvantage to asexual reproduction. So you need to know both of these methods of reproduction, and you need to know some advantages and disadvantages of each. Sexual reproduction, joining of two gametes, sex cells, sperm and egg, leads to variation. Good, because that variation leads to evolution. Bad, because you have to ensure there's a mate or someone to reproduce with. Asexual reproduction, only one parent is involved in this. That's great because you don't need to find a mate, you can reproduce yourself, but it's bad because it results in identical copies, clones, which could suffer from that lack of variation, maybe couldn't evolve, and maybe could be wiped out by a disease that affected a certain genetic code, something like that. Linked to this, you actually need to know that there are a few methods of cloning plants and animals. You need to be aware of about 4, 2 for plants, 2 for animals. Methods of cloning plants are relatively simple. Okay, cloning plants are relatively simple. My nan does it, okay, in her garden at home. She does it by taking cuttings, and cuttings are simple. If I was to take a plant, I would just cut a bit of the leaf off, and I could take that leaf and I could use a plant hormone. That plant hormone is called Auxin. You need to know that word, that's the name of the plant hormone. The only plant hormone you need to remember the name of, Auxin. And I'll probably talk about auxin and plant growth a bit later, okay? But that hormone you put on the plant, this is what's going on up here, if you can see this picture, I know it's small, but they've taken a bit of a plant and they're dipping it, the bottom it, the base of it, into auxin. And then they just put it in the ground, okay? And when they put it in the ground, it can grow new roots and it grows into a new plant, okay? So it's very easy to do. As I say, my nan can do this process. It only results in a few clones of the plant though. You get one if you do a cut-in, like one clone of the plant, okay, is what you actually end up getting. So it doesn't result in many copies. Tissue culture is the second way you need to know how to clone a plant. And it's a bit more involved. So I'll draw a little diagram of it to show, okay. Starts with a sort of similar process. You take part of a plant, maybe a leaf, okay, and you cut a section of it out. You only need a small section. So I cut a section out, and I take that section, which is a bit of tissue. Not a tissue like you would blow your nose on, but like a plant tissue. Okay? That tissue will be made up of thousands of plant cells. You get the idea. Each which have a nucleus. And inside that nucleus, it contains all the genetic information needed to make a new plant. So, using that idea that the nucleus contains all that genetic information, they actually separate this into individual cells. They break it up into the individual cells that make it up. Okay? So they break it up into the individual cells that tissue was made up of. Now, this is what's good about tissue culture. You can actually take a small section of tissue and you can get hundreds of cells out of it. As long as you're careful with them, you can divide it up into hundreds of cells. You actually take all those cells and you put them into a Petri dish. A Petri dish is one of those round dishes that us scientists like to use, that you put samples in. And you place them into there. You actually stimulate them and allow them to grow a little bit. And when they grow, they actually start to multiply. And once they've multiplied enough to give you a small amount of cells, a little ball of cells again, they can start to go into a new plant. And you can actually take a trough of some kind, put some soil in it, and you could plant all of those tissues that you have grown, all of those cells that you've grown into tiny little plants. Okay, I'm only showing a few, but in reality you could get hundreds of plants from a small piece of tissue. So that's the big advantage to it. Okay, you're going to get many clones of this plant from just a small bit of tissue. Why would you want to clone a plant? Okay, the reason you would want to clone a plant is because in just a small bit or small plant, okay, you could have a plant that's really good at, say, producing fruit. So Like a strawberry, you've got a strawberry plant and it grows the sweetest, tastiest strawberries that you've ever tasted. You want another copy of it, okay, or even several copies of it, because you're starting a strawberry business. So you can produce copies of it, and you can clone it, so you get lots of strawberries with that same characteristic. They produce that sweet fruit that you want, or they're resistant to a certain disease, whatever. Okay? They actually can make seedless grapes. Seedless grapes is a good example. Most plants grow from a seed, but seedless grapes, because they don't have seeds, wouldn't be able to grow from the seed. So they actually can do it by cloning them in some way, so you can get seedless grapes. That's an example that I can think of. So tissue culture is good because you get lots and lots of clones produced, but the bad thing about it is you need specialist equipment to do it. Not all of us can just take this tissue from a plant, split it up into cells, plant it in a petri dish with a growing medium, and then transfer it to the ground. So that's one of the disadvantages to tissue culture. Okay? You need specialist equipment to do it. Okay? So we've had about half an hour. Okay? We've got just about one question coming, and it's about tropism. Okay? So it's about using Auxin to grow a plant. I will go into it, I will talk about it, so I'll go through. If we can find out who that question's from, I'll give them a shout out. And then I'll talk about the growth of plants due to Auxin. What I'm going to do is, though, before I do that, I'm actually going to carry on with this because I want to talk about cloning animals, and then I'll come back after I've gone through the two methods of cloning animals, and I'll go into how Auxin helps plant growth. And that's a really good question because it's something that I know with my classes I haven't talked about in any detail, and that's exactly what these sessions are for, to ask those questions that I haven't really gone into, or your teacher hasn't really gone into, that you still need to know the answers to. I think it was 2 years ago there was a 6-mark question on how Auxin is used to control the growth of roots and stem in plants. So it's a good question, and we'll come to it after we've gone through the ways that we can clone animals. So cloning plants, taking cuttings, really easy, only produces one copy. Tissue culture, complicated, needs specialist equipment, but results in multiple copies of the plant that you actually get at the end. Okay? Cloning animals is more tricky, it's more difficult to do. Okay? It's not as simple, but there are two main ways you need to know for B1 for the GCSE. This is the first one, it's called embryo transfer. Okay? You could get asked a question about why would you want to clone an animal. Okay? The reasons being that you would have to give would be because you've got an animal with a certain characteristic that you like. Okay? Now in this example they're using a bull that is from a herd which has a high dairy yield, which means it produces lots of milk. So they want to produce a cow that produces the most amount of milk possible. And it's like this idea of selectively breeding animals to have certain characteristics that you want. So they want a cow that produces a large amount of milk. So they take the sperm from a bull, which produces a high yield of milk, and they take an egg from a cow, or they artificially inseminate the cow straight away. Either way, they can take the egg out and do it externally, or they can do it inside the cow. But the point is, once they fertilise the egg and they've let it divide a few times into an embryo, they isolate those embryos and they split them up into smaller embryos, still a small bunch of cells. Okay? Here it might be, say, 16 cells, and each of these might only have 4, so they'd make 4 copies, each with 4 cells. They allow those cells to divide, and they actually re-implant them into surrogate mothers, into foster mothers, other cows that are going to be carrying the offspring. Okay? These embryos, though, would not be related to this cow. They could be, it could be re-implanted in the mother, but they don't necessarily have to be. These could be any cows they were using, as long as they were healthy and able to carry a child. They plant all of these embryos into surrogate cows or foster mothers, and all of those, after the gestation period, the length of time the child develops and gets carried for, which I don't know for a cow actually, they would be born clones of the original 2 cows. So not identical copies of both of them, because it does lead to variation, because they did do fertilisation sexual reproduction, sperm and egg, fusing of gametes. So it wouldn't be an identical clone of either one of these parents, but it should hopefully have the best characteristics from both, okay, because they've taken a bull which produces lots of milk, or from a family that produces a lot of milk, and a cow from a family that produces a lot of milk, and they fuse them together to make these potentially young cows which also produce lots and lots of milk. Okay? So that's embryo transfer. It results in, these are all copies of each other, these will all be clones, but they're not clones of the original two parents, because sexual reproduction is the first step in that process. So they will have variation on these, but they will be identical to each other. In a similar way that identical twins are. They're where an embryo divides into more than one, and you get identical twins at the end of it. So that's embryo transfer, because you create an embryo, you divide it into smaller embryos, and then you implant it, transfer it into foster mothers, which then birth the new cows, or animals, whichever. This one, adult cell cloning, is slightly different. This does produce identical clones of a parent animal that you want to clone. They've done it sort of a few times, but the main one that we sort of talk about at GCSE Science was Dolly the sheep. That was the first sort of big example of where they cloned a whole animal. What they do in this process is they actually take an egg cell, an ovum, from a healthy sheep. And they take that egg cell and they remove the nucleus. Now, this you have to understand a bit of genetics, okay? You have to understand genetics for it. Inside an egg, there's only half the number of chromosomes necessary for a life. Okay, if we use the example for humans, okay, you have 46 chromosomes arranged in 23 pairs in the human cells. Okay, 23 come from your mother and 23 come from your father. Okay? When you've got a full cell, which has the 46, that's everything needed for all your characteristics, for all your cells to be the way they are. So if you take an egg, which has half the number, 23 in it, and remove the nucleus and throw that away, you're not going to need it in this process. And then you take a skin cell from another animal, same animal, but another version of it. In this example, you can see on the board, hopefully, they've used a black sheep, which they've taken the skin cell, the cell to be cloned from. And they've done that so you can see that the offspring is actually a clone of this cell, of this sheep, not this one. So when they take that skin cell, they remove the nucleus, which contains the full amount of chromosomes needed for a new sheep. And they put it into this empty egg. And they fuse the two together with a mild electric shock and some chemicals usually. And that gives you your cell, which has got all the chromosomes needed for an exact clone of this sheep. They then stimulate that to divide a few times until it becomes an embryo. And then once they've got that, they implant it into, again, a donor, into a surrogate mother, into a foster sheep, which it will be implanted in and carried until the sheep is born. Now that sheep that's born will actually end up being a clone of this sheep that actually donated it the skin cell, that the skin cell was taken from. Okay? It's not related to this one in any way, and it's not related to this one in any way. This one just gave the egg, this one just carried the baby, this one actually is the clone, the one it's cloned of, an exact copy. Okay? Problems to that are at the moment, so disadvantages, they create embryos that some might be destroyed. There's ethical ideas around it, so is it good to actually clone? I mean, it's illegal currently to clone humans, so we can't use it for that process. And also, in Dolly the sheep, she actually died fairly early because her cells were actually the age of the sheep she was cloned from. Even though she was newborn, her cells were actually older than her birth date, because she wasn't born at 0, she was born at like 3 years old, because her cells were already that old when they were cloned. So that's a disadvantage to the process. Okay. So that's cloning. Okay, I said I'd go through the two types of cell cloning, adult cell cloning, and then I'll talk a bit about Auxin, because that was a question that came in, and as I said, it's not something I've talked about too much, so it is a good question. Hopefully you can see it on this board, because obviously, because it's a question that came in, I haven't got a slide prepared on it. Okay, so I can actually put it up on the board. So hopefully, if this works, this is a new thing for us. We'll see if we can display the questions on the board. So this is us attempting to do it. So, question was actually from Rosie Tilley. Okay, well done Rosie, one of my students, obviously paying attention, and that's why she's asking it, because as I said, I'll go through it. So can you explain phototropism and geotropism? Geotropism might be called gravitropism as well. So it's actually got two names, you need to know a bit both. I do actually have a slide in it that I have shown. It doesn't actually show you too much how the process works, but it gives you the idea. Okay, trophism, if you don't know, means a plant's growth in response to a stimulus, so to happen because of something. And it's all controlled by this hormone that I mentioned called Auxin, so actually controlled by Auxin, the hormone that's in the plant. You get positive trophism, which actually moves it towards the stimulus. In the case of phototropism, that would be light. And you get negative trophism, which actually means it moves away from the stimulus. Okay? As I say, phototropism is the growth of a plant towards a light source. So if there was a light source on one side of a plant when it was growing, so this is the stem of a plant, okay, with some leaves on it. Yeah, I didn't take art, just science. Okay, and if there was a light source over here, when that plant was growing, so say the sun coming through a window, what actually happens in this process is the hormone Auxin actually moves away from light. So I'll do it in blue to try and show it, or red actually. That hormone Auxin moves away from the light and actually goes to this side of the plant. She moves away from the light source. And it actually causes this side of the plant, the growth of it, to accelerate and grow faster. So because of that growing faster, it actually grows quicker than this side, which grows slowly. It actually causes the plant to grow towards the light. Okay, so the plant actually grows upwards and curves round towards the light. Okay, so it's growing towards the light. And it allows plants to look like they're seeking out light. Now plants, obviously we know, don't have a brain, so they don't have a consciousness to go, oh, there's the light over there, we'll go towards it. But because the Auxin moves away from the light, okay, because the Auxin moves away from the light and actually stimulates the stem to grow quicker, it actually causes this side of the plant to grow quicker than this, and it actually bends towards the light. That's called phototropism, because it's growing towards light. Photo, meaning light, and tropism, to grow. Ooh. Okay. So that's growing towards light. Now, that only happens in the stem, really. You actually get tropism in the seeds as well. So probably put it on another piece of paper, because otherwise I'll run out of space. No, I'll do it down here. So this is gravitropism. So you have a seed. When that seed first grows, okay, the root comes out of one side, and the stem comes out the other. Now if they keep growing sideways, or whatever way they've fallen, that's not going to be very helpful to the plant. So in gravitropism, or geotropism, okay, geo meaning earth, Auxin is affected by gravity. So Auxin, I'm still using red for, it actually falls to the bottom of the plant. Okay, so it sinks to the bottom because gravity pulls things down. Okay, so all that Auxin sinks to the bottom of the root and the stem. Now, just like before, in this example, the auxin actually causes the stem to grow faster. And because that side of the plant grows faster, it actually causes it to curve upwards and then start to grow leaves. Okay? So it will actually grow upwards, away from gravity. Whereas in the roots, auxin actually causes the root's growth to slow down. Okay? And because it causes the root to slow down its growth, it actually means the top grows faster, and again, it then grows downwards. Okay? And starts putting out little roots. Okay? So the growth of there is downwards. So auxin sinks to the bottom, causes the stem to grow upwards. So it makes the stem grow faster, and auxin makes the roots grow slower. Okay, and that's what actually causes it to bend up as the bottom of it grows faster, curves up, and then grow down as the bottom grows slower, it curves down. Okay, this is really a high level topic. I mean, the ideas about auxin and knowing auxin as the plant hormone, we should know that. So we need to know that the hormone involved in plants growing is auxin. We need to know that it grows towards light in phototropism, and in geotropism the roots grow down and the stem grows up. Okay, those are the basic bits. Okay. The detailed bits is this bit, where auxin moves away from the light and then causes the stem to grow faster, so it bends towards the light, and it sinks down in the stem and the root, and that's what makes the root grow down and speeds up the stem so it grows up. Okay? So that's the detail of it. The basics are just here. Auxin can also be used while we're on it. It can be used as a weed killer, so you can actually use auxin to make some leaves grow out of control, or some plants with broad leaves grow out of control to the point where they can't sustain themselves and actually die. So you can use it as a weed killer. It can be used as a rooting powder. Now that's in taking cuttings, so that's what I talked about when I said take cuttings. You actually put auxin on the plant and plant it in the ground, and it makes the roots grow out. You can actually use it to make seedless fruit, so flowers sprayed with the hormones to make the fruit develop but not the seeds. So you can actually plant the plant, and when it starts to grow, you actually cause the fruit to grow rapidly, and it doesn't actually develop a seed inside, so seedless grapes again. And you can use it to ripen fruit. So if you've got a fruit tree and the fruit isn't ripening up, or you want it to ripen faster so you can sell it, you can spray it with hormones such as auxin to encourage the fruit to ripen, so you can actually grow it. So hopefully, Rosie, that answered your question. And you can see now how auxin actually controls phototropism, growing towards light, and geotropism, roots growing towards gravity, stem growing against gravity. Okay, so that's auxin and its effects on a plant. So that was a question we had come in, and hopefully I've answered it in any detail. But if you have any more questions, Rosie, then do email back in, or if you want any more information, I'll try to come back to it. But that's kind of what you need to know anyway. That's as detailed as you'll be expected to get, even in the 6-mark question I did see come up on this topic. Okay. Remember, if you do have any more questions, do email them in to the communications email, and I can put them up on the board, and I can have a go at answering it for you. Going back to where we were, okay. A question has come in about decay, so I will go on and do a bit about decay, but just because it's linked to what I was talking about before, about different types of cloning and inheritance, and it's what I think, alongside this with auxin, probably one of the toughest topics in B1 that can come up, is to do with genetic modification, and I don't think it's ever taught in any great detail, so I just want to go over the points I think you need to know about it. So I'm not going to go too much into it, but I'll go into the points that you kind of need to know and recognise. Okay? Most of the time, when we talk about genetic modification, we're working with bacteria. Bacterium is just a word for a single bacteria cell. Okay, bacteria are special because they have plasmids in them. Plasmids are like rings of DNA. They're circles of DNA. And I said a section of DNA was called a gene. So a small section of DNA is called a gene. So if we've got this plasmid inside a bacterial cell, we can actually extract it, take it out of the cell. And you can actually cut a section of that DNA away. It's like using scissors to cut it out. Okay, so you take a pair of molecular scissors and you cut out part of the plasmid. So a plasmid is that circular bit of DNA. And a small section of DNA is called a gene. So we cut out a gene, like using scissors. And again, I said I'm not an artist, so there we go, there's my molecular scissors. Now obviously these aren't real scissors because scissors wouldn't do the job because we're talking about DNA, it's tiny, okay? So they actually use, these scissors are enzymes. Yeah. So these scissors are enzymes and they actually extract or cut out this section here of DNA, small piece. They can then replace that bit of DNA in the plasmid with a gene that they want. So this is the human gene for insulin production. So they actually take the gene out of a healthy human individual, they extract some of the DNA and they cut out, they've isolated just the gene for making insulin. And obviously if your cells don't produce insulin, if you've researched hormones, you know controls blood sugar. And people who are diabetic don't produce enough insulin in their pancreas. So they have to take insulin injections. And to do that, we need to produce the insulin for them to take their insulin injections. So they would cut out that section of DNA using an enzyme, and then they would insert that gene that they've cut out of some human DNA from using enzymes into the plasmid. That plasmid can then be inserted back into a bacterial cell. And then they can stimulate the bacterial cell to divide. And when it divides multiple times, they have this bacterial cell which can actually produce human insulin. So it actually makes insulin because it's been given this gene that's responsible for producing insulin. They can then harvest this insulin that's produced and then that can be used to give insulin injections. So that's one way of genetically modifying a bacteria. Now the main bits are, you need to know that the DNA is cut using an enzyme. It doesn't have to be in bacteria. You could, and people try to do this, genetically modify a plant. People talk about like GM plants and problems with that. So if you took a piece of DNA, okay, of a plant, you could actually take your enzyme again, and you could cut out a section of DNA, and you could replace it with the gene that was resistant to weed killers. Okay, so that gene that we've got there is resistant to weed killer. Okay? We actually, splice is a good word. Splice means you just insert it into the DNA. So you cut it out using an enzyme and you splice that gene into the DNA of this plant. So your DNA now has this resistant to weed killers section of DNA in it. You then grow those plants, and those plants, because they have this gene which is in their cells, are highly resistant to weed killer. And that's great because now when you have your crops, you have your plants, you can spray weed killer all over them, kill all the weeds, and the plants won't die because they've got this gene which is resistant to weed killer put back into them. The problem with that is if this gene never got out into the wild flower population, all those flowers would also become resistant to weed killer. And if you want to kill, if some of the weeds that you're trying to kill actually end up getting this gene, they absorb it into their DNA, then they could become resistant to weed killer. And then you would have a major problem because your weed killers wouldn't work on the weeds you are trying to kill. So you were spraying your fields, your crops with weed killer, and all the weeds would be resistant to the weed killer you were using. And they would take all the nutrients, all the goodness, all the light that was designed to be for your plant that you were trying to save in the first place. It's why there's issues around genetically modified plants, because it can escape into the wildflower population. Another thing it could do is it could also, we don't know the effects. If that was a crop that we were growing, say flour or maize, that we were using for food or to make bread, we don't fully understand the effects of that gene on the human population. And therefore, we don't know if it would have harmful effects to humans. Okay, so we're not aware if this gene has harmful effects. So that's a big thing against genetic modification, or genetic engineering as it's sometimes called. Okay, we don't know the full effects of those genes that we're putting into other plants and animals. So that's a concern. Well, until that study has been done, it's probably unsafe to do. So that's why there's a lot of opposition to it. You can have, again, the usual arguments of the opposition, as in it's against religion, but you have to bear in mind, like I said earlier with that argument, you have to say, why is it against religion? Because people don't like the idea of playing God, or creating a new species, or meddling with nature, messing up with nature. So try to avoid it if you can think of other reasons, such as the gene could escape into the wild animal or plant population. We don't know the effects of that gene on the human body if we're going to eat that crop afterwards. So that's something to definitely bear in mind. Right. I might skip ahead because a lot of questions coming in. Well, not a lot, but a few are coming in around decay. So I might jump ahead to decay, the decay process. It is an interesting one. So I'll go straight into decay. And decay, you get questions around what is used for decay, how do you control decay, how do you speed it up, what's its uses. So this is the decay process, okay, summarised here. This should be in your revision guide, so you may have seen it before, but a decay process is when an animal or plant dies. When an animal or plant dies, okay, those animals and plants will fall to the ground. Now this is provided we're not talking humans here and necessarily they get buried or cremated. We're talking about in a normal ecosystem, so in a forest somewhere. Plant animal dies, it falls to the ground and it stays where it is. The first thing that's going to break it down is detritus feeders. That's worms and maggots. They start eating it, okay? If you, I don't know, seen, there's plenty of videos. They did a thing on Mythbusters where they took a pig and they left it somewhere for a while and saw what happened when it rotted over time. The first thing that's going to start eating away at it is worms and maggots. They're going to break it down. And they take all the nutrients that they need out of the plant or animal that's dead, and they actually produce waste material. So essentially they poo out what they don't need. Okay? That's already partially broken down. And on the carcass that's left, the substance that's left, what will actually happen is microorganisms will begin to grow. Microorganisms are bacteria, and not viruses in this case, but moulds and fungus starts to grow in it. They grow in it and they start breaking down the cells into the components they're made up of. So they break it right down into the nutrients that actually make it. Okay? And when they do, they take in some of the nutrients and they grow, and then they reproduce themselves by dividing. Okay? So more and more bacteria, mould, fungus grows on it, and it just breaks it down more and more and more, until eventually you're left with a soily-like substance. And that soily-like substance is all the waste products from the microorganisms. It contains nutrients and lots of water that's been broken out of the cells, and it also released CO2. And the reason it released CO2 is a process we haven't talked about, but is so important. There's two processes you should know for all biology, really. Jump to that. One of them I'm sure you'll recognise, and that's this, carbon dioxide plus water uses sunlight and produces glucose and oxygen. Okay, that's a process I'm sure you'll recognise as one that goes on in plants called photosynthesis. Photo, again meaning light, and synthesis to make something. Okay, and then simply put, the opposite to that process is respiration. So you get respiration, which is exactly the opposite. And this is so important, this process, because every living thing does respiration. Even plants that do photosynthesis, they'll also do respiration. The equation is just the same as that, just backwards. So glucose plus oxygen goes to carbon dioxide and water. It doesn't matter the order of these things, only they're on the right side of the arrow. So glucose and oxygen must always be over here, but you could put oxygen and glucose, doesn't matter. Carbon dioxide and water must always be over here in respiration. Doesn't matter if you've got water and carbon dioxide. Similarly with this, doesn't matter the order of that and that, but they have to be on those sides of the arrows. The reason I talk about that is because those decomposers, those microorganisms, they are living things. So in the decay process, they do respiration. And when they respire, they need oxygen and they break down glucose or sugars in the thing they're breaking down, they're decomposing. And they release carbon dioxide and water, and that carbon dioxide goes up into the air. And this forms part of the carbon cycle. This is one part of the carbon cycle. Okay. They also, when they release those things, those nutrients and those minerals go back into the ground. Okay. And when they're in the ground, and we're talking about, I mentioned a forest at the start, the forest can actually grow using those nutrients. So the plants will take in those nutrients when they grow. They also will take in CO2, carbon dioxide, from the process of photosynthesis. So the microorganisms are releasing it in respiration, and then plants are taking it in when they grow. So it's a cycle. These things cycle round. And then when those plants that have grown have been eaten by animals, and those animals have died, or the plants have just died, the decay will start again. Detritus feeders, worms and maggots, decomposers, microorganisms, bacteria, moulds, fungus, release the products, the nutrients, back into the soil, and it just keeps going round. So it's like a cycle. The main sort of points I've tried to summarise over here. Microorganisms are responsible for the majority of decay. So yes, worms and maggots are part of the process, but when they're asking you questions about it, it's really microorganisms they like you to talk about. Okay, they like you to talk about microorganisms in the main. Bacteria, moulds, fungus, not viruses in this case. And you also need to know ways that this process can be sped up. Now, microorganisms being living things, they actually like it warm. Like us, they like it about 37 degrees Celsius. Okay, so 37 degrees Celsius, they like the temperature, so they like to be kept warm. They like it moist or damp, which just means they like some moisture, they like water. So anything you can do to increase the amount of moisture or keep moisture in is going to speed up the decay process. It helps them break things down. They also need it for their cells. And because they're doing respiration, just like us, they need lots of oxygen present. Questions on it may come up. I've seen this a fair few times where a gardener is building a compost heap and he has his compost bin has like holes in the bottom of it. Okay? And these holes are for something. He piles up all the waste materials, get piled up inside the bin. But there's actually holes that I've probably made too small, but are in the side of the compost bin. And the reason those holes are in there, the holes are to allow oxygen to get in. Oxygen can enter, and the microorganisms need that. And it can also let heat escape, because you don't want to get too hot in the compost heap. Okay, because then it will start to kill the microorganisms. They can only take a temperature, like we can, of up to just over 37 degrees. So, very badly drawn, I know, but this is my compost bin, and this is my compost being made in it. And in there are microorganisms breaking down all the food waste I've put in it. There's holes in the bottom to allow oxygen to get in and allow heat to escape. I was looking at an exam paper with someone the other day that talked about a bird that actually had its nest, built its nest, and it actually filled up its nest with grass cuttings, so bits of grass, dead grass that it pulled out. And then it built sand over the top and put like sand blocks with little holes in it, made a dome of sand over the top, and it actually laid its eggs inside the dome itself with grass cuttings underneath. And then it didn't need to sit on the eggs, so it didn't need to sit on its eggs. It could just leave them in that little dome that it built. So normally birds will sit on their eggs, as you probably know, to incubate them. But in this case, this question that was asked, this bird didn't sit on its eggs. It put grass cuttings underneath this dome that it made of sand and laid its eggs inside. That was because the grass would decay, because microorganisms would get in, and when it decayed, it would give off heat. It would get warm through respiration. So that would keep the eggs warm, so it didn't need to sit on the eggs. Now, it also talked about, it went on to ask, why did the bird open these holes in the day and close these holes at night? It would open the holes to allow moisture and oxygen to get in. And it closed the holes at night to make an insulated blanket to stop the heat escaping. So at night time it was warm. So that's a really clever adaptation of this bird. I can't remember what the name of the bird was, but it was actually in a GCSE paper, I think from 2014, possibly 2015. Okay. So if you want to have a look at it, like I say, June 2014, maybe 2015 was the paper it had that question. So that's kind of an overview of decay. Okay, and they like asking about compost heaps. Another thing you can do with a compost heap is you can actually dig it and turn it over. And when you do that, you often, if it's a cold day, you'll see steam rising out of it because all the heat is released, and that's to stop it getting too warm inside, and you're allowing oxygen to get into it. So that's an important bit as well. Okay. We've had some more questions come in, I believe. We'll get to those in a bit. We'll go back and do a little bit more because we've got about, well, just over 20 minutes left of the stream. Hopefully we've covered some topics that you've been interested in, but if I haven't, or there's something you're really desperate for me to go over and I don't look like I'm talking about it at the moment, then that's what the email address is for, to email in that question and then I'll go through it. So I'll be doing about another maybe 10 minutes at the most, and then I'll try to go through some questions at the end, and then if I've got any other time I'll just go into some random little bits, just bits to remember, little bits to remember, little tips. So, where were we? We've done decay, genetic modification. I wanted to go into a little bit, because it comes up a lot, but of healthy diet. Now, healthy diet is a topic that students tend to be very good at, so that's why I'm not going to go into the details, but it does come up a lot, so it's worth knowing a bit about. You need to know the main components of a balanced diet. And by the main components, I mean these three really. Carbohydrates, and you need to know what they're for. They're for providing energy, for respiration. Carbohydrates are glucose for respiration. You need to know fats, which can also provide energy, but it also makes the cell membrane, which is the outer layer of cells, and also, like me, it insulates you. So it insulates you and can keep you warm. So that's what fat can be used for. Protein is used for growth and repair. So it builds muscles, it builds cells. Okay, that's what it's used for. And these are the three, carbohydrates, fats and proteins, the three main components you need to know for a healthy, balanced diet. The other one that occasionally comes up is vitamins and minerals. You need to know that vitamins and minerals are important, but mainly about those, you just need to know they're important for life processes, to keep the cells functioning. You don't need to know details really of them. Those are the ones I see come up. So you need to talk about what is a healthy diet and why is it important. And I'm only going to briefly say that you should know that if you're overweight, it can lead to heart disease, can lead to diabetes, are two good answers to give, problems with being overweight. and being underweight, that's a really interesting one, because I said to my classes and I said to people at Saturday school, I see so many times students put in, what's the problem with being underweight? They say anorexia, and that's not correct. Okay, anorexia isn't a problem of being underweight. Anorexia leads to you becoming underweight, so it's not accepted as an answer. So if it is a question about what is a problem of being underweight, you can put becoming malnourished. You could put a specific example of being malnourished. Malnourished means not getting enough nutrients in your food. A specific problem, if they ask you, give an example of being malnourished, could be scurvy. Scurvy is a disease that sailors used to get when they didn't have enough vitamin C and their teeth used to fall out. You could put lack of energy is another problem with being underweight. Okay? So that's balanced diet. Whether you're under or overweight can be affected by your metabolic rate. And the metabolic rate is how fast the chemical reactions in your body actually take place. So, metabolic rate. It's the rate of chemical reactions, or speed of chemical reactions in the body. Okay, rate of chemical reactions in the body. Particularly respiration, so burning your food, but could be any of them. Okay, and you can inherit it from your parents. It is affected by your age, by your gender, and it's also affected by how much muscle to fat ratio you've got. Okay. Pathogens, a little bit on it. So a pathogen, a definition that you need to know, is a pathogen is any microorganism which causes disease. Okay, so a microorganism that causes disease is a pathogen. The two main types are bacteria and viruses. You don't need to know much detail about them, other than they are both pathogens. It doesn't mean all bacteria are pathogens, only the ones that cause disease. So bacteria can be killed by antibiotics, whereas viruses cannot be killed by antibiotics. Viruses tend to live inside your cells, so are very hard to kill, so that's the important point. They can be spread in four main ways. Okay, you've got droplet infection, which is where if I sneezed on this video, you'd probably see me spray out a load of little droplets into the air. Wouldn't be very nice. And those droplets can be inhaled by other people, and they contain microorganisms, or pathogens I should say, that could cause disease. Direct contact, so if someone's got a skin infection and they touch someone else, that can be directly transferred to their skin and cause them problems. This may not be clear on the picture, but it's actually a little cut in someone's hand, so a break in the skin is a route for pathogens to enter the body. And this is if you eat contaminated food or drink contaminated water, it can lead to a disease that will affect your intestines or stomach or something to that effect. Okay. They're the four main ways you need to know that they can be spread. The body can prevent itself from a lot of these by, your skin is actually a barrier, so your skin protects you from pathogens. You actually have millions of them on your hand at any one time, but your skin is protecting you from them, so they're not actually making you ill. They want to get inside your body. Cuts. Scabs will form on them. So if you get a cut, you'll get a scab, and that will actually cause it to block any pathogens getting into your body system. Droplet infection, so if you do inhale some microbes, your airways, your throat and your nose have mucus in them, which the pathogens get stuck to, and that means you can actually stop them getting into your lungs, where they can cause you a lung infection or some problem like that. Those are physical barriers, ways that you prevent the microbes getting in, but that's not always good enough. Occasionally a microbe will get into your system, and if it gets into your system, you have a chemical defence mechanism, which is white blood cells. White blood cells, they're not living things, although we often think of them as living things, and you can't describe them as a living thing in an exam. They're parts of cells, or part of your body, they are cells, that actually ingest pathogen and destroy them. So that means they actually engulf, they like eating. So if this is the pathogen that's inside your body, Okay, a white blood cell comes along and it actually goes around it and it ingests it. It doesn't eat it because it's not alive and it doesn't get nutrients from it necessarily. It just goes around it and breaks it down. Okay, so that's one method it can do. It can ingest them and destroy them. It can also produce antibodies. So it can produce antibodies which actually stick to the surface of it, and those antibodies stick to the pathogen and they can kill it or render it harmless. Okay, so it can produce antibodies. Antibodies and antibiotics that I mentioned earlier are different things. Remember that. Antibodies are produced in the body. Okay, antibiotics are drugs that affect, that kill bacteria but not viruses. So produce antibodies to destroy a particular pathogen. And you can also, they do also produce antitoxins. And antitoxins just stop the harmful effects of the bacteria, so stop you feeling sick from it. Okay. Bacteria, if we use too many antibiotics, different to antibodies, can become resistant to antibiotics. And the example they sometimes talk about is MRSA. That's a disease in hospitals that's caused by a bacteria that has now become resistant to antibiotics, meaning it won't be killed by antibiotics. Okay. The way it works is if antibiotics are overused or you don't complete a course of antibiotics, it actually kills all the weak version of the bacteria that's in your body. The antibiotics kills the weak versions and the strong ones survive. And those strong ones actually go on to reproduce. And because there's no competition for them anymore, because you killed all their competition, you killed all the weak ones, and you stopped taking the antibiotic, they actually reproduce all just their offspring, which are all strong. And because they're now all strong, okay, they can no longer be killed by the antibiotic. So now you've got a much harder to fight off infection. Okay, and this could be a big problem. As I say, it's happening because in the past we've overused antibiotics and they were prescribed by doctors for just about everything, but also people didn't finish the course. They'd take it for a couple of days, feel a bit better, so stop taking it. And the last thing before I go to some questions is just about vaccination. So you can be vaccinated, and some people in the school have had vaccinations recently against the disease. And the way they do that is they inject a weakened version of the microbe, the microorganism, the pathogen, into the body. And the body actually fights it off. And when it does, the white blood cells produce antibodies. Remember, these are antibodies produced by the body, these things. Okay? They then kill that weak version of the virus very easily because it's weak and it can't put up much fight. Then the body leaves those antibodies in the blood. So those antibodies stay in the blood. And it actually remembers, the next time that virus comes into the body, it actually remembers that infection and it then can destroy it really quickly before you feel sick. One they do talk about is MMR. So the MMR vaccination, which stands for measles, mumps, I think that's spelled, and rubella. Okay. It's actually a vaccination that's given to young children to prevent them getting all three of these, which could be very dangerous diseases. So they give you a weak version of all three, your body fights them off, the antibodies are in your blood, and that remains there for if you get reinfected. Okay. So we're coming to the end of the topics I was going to talk about. I believe there's been a few questions that have been emailed to me. So I'm going to take a little look at some of those, try to answer some of those in the last sort of 10 minutes that we've got left. Hopefully you find it useful. And there's be a few little tips in this last bit as well that you should be able to get. Just remember, please subscribe to the video because we will be doing streams for the chemistry exam, the physics exam and the additional exams after half term. And there are notes available on the Science Revision website if you want to read up on any more of these topics. And I have to say I haven't covered everything in B1. There are still a few things like energy and biomass, more details of inheritance. I mean, the majority of it we've got through, but energy of biomass is definitely one that you might want to take a look at, definitely. So, let's just get up this question, find out who it's for. Right, so good question by Emily Martin. So we've got, can you explain the hormones of the menstrual cycle? So the menstrual cycle is obviously a cycle that takes place, they say every 28 days. Once a month is a way of remembering it, but if you get a multiple choice question on it, it's 28 days. It's the reproductive cycle in females. And yeah, unfortunately guys as well, you need to know about this because it can get questions on it in the GCSE. It doesn't come up too often, but it is a tricky topic when it does. So looking at some of the specific hormones in the menstrual cycle, this table, which is in your revision guide, shows it pretty nicely. Okay, you've got FSH, which is follicle stimulating hormone, it stands for. You can remember it, Ms Charbonneau told me this by the way, because it stimulates the egg to mature. So follicle stimulating hormone. It stimulates the egg to mature and the ovaries to produce estrogen. So it does those two things. So in the ovaries there are eggs and they mature when it senses follicle stimulating hormone. It's actually produced by the pituitary gland, which is a little gland at the base of the brain. Okay, so that releases it. It goes into the blood. And this is a good point about nervous response versus hormone response. Nervous response happens quickly, okay? You can do things really fast. Hormone response takes a lot longer because it travels around in the blood. It's not an electrical signal, so it goes around slower, okay? So it's injected into the blood in the pituitary gland or by the pituitary gland, travels down till it gets to the ovaries and it stimulates the eggs to mature and starts the production of estrogen. Estrogen is the one that is usually thought of as the female hormone. Okay, it's made by the ovaries, and that actually causes the lining of the womb to become thick and get ready to receive an egg. So the lining of the womb or uterus thickens and actually becomes ready to accept a fertilised egg. And most of these things do another thing as well. They kick on the next stage of the process. So it actually starts, the pituitary gland, at the base of the brain, start to make luteinizing hormone, LH. I don't really have a clever way of remembering that, luteinizing hormone, but LH is fine, you can call it that. When it's released by the pituitary gland, by the time it gets down to the ovaries, it actually causes the release of an egg from the ovary. So follicle stimulating hormone doesn't cause the egg to be released, it just causes the eggs to start to mature, to get ready to be released. And it's actually luteinizing hormone that causes it to be released. Now, the last in this sort of process is progesterone. Now, progesterone is produced by the ovaries, and it's actually released if the egg becomes fertilised, and it causes the lining of the womb to stay thick, the lining of the uterus to stay thick, if it needs to accept a fertilised egg and hold it through gestation till the baby is born. The reason this is a good one to know is because it's used in the contraceptive pill to keep the lining of the womb thick and actually stop the process. So the body is almost fooled into thinking it's pregnant, and when it thinks it's pregnant, it's not going to go through this process again, and it's not going to release another egg. So it's used in the contraceptive pill progesterone to keep the lining thick and stop the release of any more eggs, therefore stopping pregnancy. Okay? If you're talking about the IVF and fertility treatments, you actually use these hormones because you'll use follicle stimulating hormone to stimulate the eggs to mature, and then the eggs can be removed via a needle, and then they can be fertilised outside of the uterus, outside of the womb. And then once they're fertilised and have divided into an embryo, they use these other hormones to make the lining of the womb thick and keep it thick, and they re-implant the egg back into the uterus. Okay? So these are also used in fertility treatments. Fertility treatments can be good because they allow people who otherwise haven't been able to have children to have children, but there are issues with them. It used to be a big issue of multiple births. You could have more than one child born. They also could lead to issues because they're expensive, so that can be a downside to them, a disadvantage to them, and they're not always successful. Okay. We have a couple of questions. Pyramid of biomass, and can you explain biomass? So this is what I was talking about, the only real thing I haven't covered, so you've asked me about it now. And since we've got 5 minutes, we'll go into it. Okay. Biomass. It is the dry living mass of an organism. So that's what biomass actually is. It's the dry mass of a living organism. Okay, so that's what the biomass it actually is. Okay, it's what it would be is if you took me and you extracted all the moisture from me, so took all of the moisture out of my cells, and you're just left with a pile of carbon, because that's pretty much what I am, and a few other elements, then you would have my biomass, and you would weigh it, and that would be the total amount of mass that makes me up. That is essentially what I've used to grow into the incredible human being I am today. Okay, so it's all my mass that makes me up, and in that's passed up through a food chain. Biomass is the substance, the matter that you actually use to grow. Okay, so as I said, biomass is the dry mass of living material in an organism. It's in a food chain. So when you have a food chain, it always starts with what's called a producer. So every food chain starts with a producer. And why it starts with a producer is because a producer produces its own food. It makes chemicals from the sun. I'm just going to have a little drink because my throat by this point, and we'll be going for about an hour and 20 minutes or 25 minutes, getting quite dry. So the plant, the producer, produces its own food from the sun's light through photosynthesis. I put the equation on the board a while ago, but not going to get it back, but hopefully, carbon dioxide plus water goes to glucose plus oxygen. So it captures the sunlight, makes its own food, and stores it in itself. And it uses that to grow. So it grows bigger, the plant grows. Then an animal eats that plant. When it eats that plant, the first thing it does is it probably doesn't eat all of that plant. It might eat the leaves, it might eat the fruit, but it probably doesn't eat all of the plant. And that means some of the biomass is left behind. So this insect, in this example, already hasn't taken all of the biomass from the plant. It's losing biomass. It's losing some dry mass as it goes along. It then will move about to find more food, move about, and when it does, it respires. And when it respires, it gets warm, so it also gives out heat. So there's heat loss. It also has to excrete some things, so it's lost in its feces and in its urine as well. It actually loses energy. So in this example of a cow, it takes in 3,060 kilojoules of food. 1,110 kilojoules is lost through heat, and 1,797 kilojoules is lost in the urine and feces of the cow. That only actually leaves a very small amount, around sort of 200 kilojoules, maybe less, 100 kilojoules, which it can use to grow. And when it uses it to grow, that's when it increases its biomass, by actually using it. So it's losing energy in multiple different ways. And at every stage in a pyramid, energy has to be lost. I'm going to use a different example to that for a food chain. So mine, I used it in lesson today, is actually a tree getting eaten by a caterpillar. I think this is a good example because it shows it nicely. Getting eaten by a bird. Okay, if I was to do what's called a pyramid of numbers, that's my food chain, the arrows go that way because they show the energy moving up through it. If I'm to do a pyramid of numbers, that would have quite a strange shape. The reason being is one tree could sustain thousands of caterpillars. Okay, just one tree could have thousands of caterpillars on it. And those thousands of caterpillars could feed 100 birds, maybe. Okay, so that's my tree. You have to label these. That's my caterpillar. And that's my birds. And that's pyramids of numbers. if I was to do a pyramid of biomass, pyramids of biomass always have to have this shape. Okay, they always have a pyramid shape, so they're easy to draw. Because if I dried out that tree, took just the dried weight of that tree, its weight would be huge. Okay, it would be massive, that would be the tree. And then if I took those caterpillars, even though it's one tree and it's thousands of caterpillars, and I dried them out, they would weigh significantly less than the tree. And then similarly, if I took the birds and dried them out, they would weigh less still. So my pyramid of biomass always has a pyramid shape. Okay. And the reason for that is energy is always lost as you go up. So energy is always lost as you go up your pyramid. Okay? Because of the reasons we've said. Because you lose it in heat, keeping your body warm, you lose it in movement, moving about, you lose it in respiration, you don't eat all of the animal underneath you, so even when this owl eats this mouse, it won't digest the bones of the mouse, so they're already going to be lost, and it will also excrete some other waste products that it won't take from the mouse. Okay, that is our hour and a half. Okay, we've gone through just about everything. As I say, I didn't go into detail on lots of topics. If there were some questions, I tried to answer those. You can find all the information. You should have a revision guide at home for tonight. There is the website, which the password, if you don't know that, is Year 11 Folkestone, all one word, all lowercase, to see some of the slides. And then you can basically do revision tonight and try to do as well as you can in your exam tomorrow. As I say, we will be doing these streams for the C1 exam and the P1 exam before half term, and then the B2, C2 and P2 after half term. If you have any suggestions for me, just come and see me, tell me what you liked, tell me what you didn't like, and I'll try to make it better as we go on and do more and more. Hopefully you found it useful, and the only thing that's left to say from me and all the science team is good luck tomorrow, and we'll see you before. Good night. And we never quite know if it's finished, so I might still be live. If I am. Good night. Again.
Just turn it off.
You're shaking your head now.
You give me the password.
You've given the password to everyone on there. But do you know what, if they just sat through 90 minutes of you talking, they deserve the password.
That's their payment.
Was the thought that, you know...
Who cares?
Yeah. I have already put up...
Like I say, it's only going to be live for another a few more weeks.
It's still live. I don't know why it's still live. They could be recording our conversation right now.